Okay, hello and good afternoon, good evening, good morning, depending on where you're joining us from. Welcome to the Twist Bioscience webinar, Connecting Plant Trait Genotypes to Phenotypes using whole genome sequencing or Skim-Seq. My name is Paul Doran. I'm in the business development group here at Twist. Before we get started, just a few items to go over for housekeeping. First is we're going to answer all questions after the presentation, so if you could hold them till then. If you have a question, please submit it to the Q&A, which is there's a button down on the bottom. Following the presentation, if you have a minute, we'll have a brief survey. We'd really appreciate your feedback. And before we kick off with our main speaker today, I'm going to give a kind of a brief overview of some of the things that we're doing at Twist that's relevant to Jason's presentation. Okay, so we'll do the overview, introduce Jason, we'll do the Q&A. So what's unique about Twist Biosciences? It's really our DNA writing platform. Our technology enables us to make an enormous amount of high quality DNA at scale. So if you look at everyone else, they're typically doing one oligo per well, whereas at Twist, we can make in the neighborhood of about 1 million oligos per chip, which gives us enormous capabilities to do some of the following. And so if you look at what we're doing, we produce genes, we produce oligo pools, we make variant libraries for screening. And then relevant to this talk, we make oligos for doing NGS. And then on the biopharma side, we're helping people make vaccines. We're making vaccines and doing antibody development. And then lastly, which I think is pretty cool and exciting, is we're leveraging our ability to write DNA for data storage. And that sparked collaborations with the likes of Netflix, Microsoft, and companies like Western Digital. Okay, so the technology that Jason used to do screening for his for the presentation that's coming up is a product called Twist 96-plex. So I'm just going to give a quick overview of it. So it's a library prep that's fundamentally different than other library technologies. There's no fragmentation, end repair, or ligation. It's tunable for high and low GC content. And there's little and no automation needed. And so you can also do 96 to 960 samples at a time. Okay. So how does it work? So you take your DNA template and you put it in a 96 well format, and then you add random primers. So depicted in green is a random primer followed by a sample barcode, followed by a partial P5 Illumina adapter. These will then hybridize to your template, and then we do an extension reaction, which will then be terminate with dideoxy that's biotinylated. That will produce this fragment right here. Now that you've got your biotin, you can capture that molecule. So we do a bead capture. We wash away anything that doesn't have the biotin. Then we come back at the next step with another random primer, followed by a partial P7 adapter. We extend that out with a high fidelity strand displacing polymerase, which then produces a fragment that's got both the P5 and the P7. We come back with a PCR, and that essentially would produce your fragment. The PCR is it's just a few rounds of amplification. And the final step is to do a size selection, and then it's ready to go into the sequencer. So what we're doing next with the 96-plex, we're doing some development work, which we're really excited about. We've got some preliminary data that's looking really promising in terms of using this to do capture. So basically what you do is you produce a 96-plex library, and then instead of just doing your standard low pass, we run it through our protocol that's been slightly modified that allows us to do target enrichment. After you do that, you can either use that for targeted sequencing, or you could blend the two together to basically enhance areas of the genome that you want to take a look at. Okay, so my last slide here before introducing Jason, I just want to kind of go over some of the applications of the 96 with low-pass whole genome sequencing. And I put this up here just to kind of help clarify. There's a whole bunch of different names for low-pass or Skim-Seq. So I threw out some of the ones that I've heard. You've got genome skimming, low coverage, Sparse-Seq, shallow-Seq, and if people know of other things that it's called, please put that in the chat/Q&A. So some of the applications for this assay, QTL analysis, which is what Jason's going to be talking about, and mapping populations. You can do GWAS, you can do diversity analysis and population stratification. You basically do SNP and or KMAR analysis, basically where you use KMARs as a marker. You can do fine mapping SNP discovery, actually both at the same time. Looked at structural variation, such as deletions, insertions, and rearrangements. It works with and without references, and it utilizes new reads. This is a paper that we put out from work that I did back in 2018, where we were just looking at, comparing this to array technology, and we were essentially able to replicate a 600K array with 0.01x coverage in corn. And so it's been pretty exciting to see how much this approach has really kind of taken and becoming really popular over the last probably like 4 to 5 years. Okay, and with that, I'd like to introduce our main speaker for today, Jason Johns. Jason is a PhD candidate at UC Santa Barbara, where he studies the genetics of Alpine adaptation, floral development, and population genomics. He earned a bachelor's degree in business administration at Cal Poly San Luis Obispo, and worked in various capacities in the wine industry after graduation. So he later returned to Cal Poly to get a master's in biology, where he described a previously unwritten pattern in plant in cook pines, which is cultivated worldwide. Trees in the northern hemisphere lean south, and trees in the southern hemisphere lean north. Jason has enjoyed teaching several classes during graduate school, including lecturing the introductory genetics course at University of California, Santa Barbara. Going forward, he plans to contribute to a broader understanding of molecular plant mechanics to facilitate our management of both food and biodiversity. With that, Jason, over to you.
Thanks for that intro, Paul. Nice to get some context for Twist and the assay that I will be going over here. So I'll start by thanking all of you for taking the time to be here. And I'd like to thank Twist for giving me the opportunity to talk about some of the work that I've done over the last few years. So as Paul mentioned, I'm wrapping up a PhD program here at UC Santa Barbara, and this project that I'll talk about here today represents an exciting piece for me of my work. So our lab has mostly used columbines, pictured here, to study evolutionary genomics. And so that's the context I'll be using to describe this method. But as Paul mentioned, this method is applicable to many systems examining the genetic basis of a trait. So before I get into the details of why and how we made a genetic map and did a QTL analysis, I'd like to just briefly contextualize my research. So most projections anticipate that we'll have about 10 billion mouths to feed by 2050. And so in that anticipation, there have been all kinds of innovations in agriculture. Technological innovations like using drones to monitor crop fields, mechanical innovations like self-driving tractors, spatial innovations like going vertical with growing, and of course, innovations in breeding for a long time. So not only is food shortage a threat, but so is the impact that growing that food has on the health of our ecosystem. And so we're currently doing a dance trying to strike a balance between agricultural efficiency and caring for the planet. So as we more and more realize the ecological impacts of decades of large-scale worldwide agriculture, there's been a movement toward ecological awareness in agriculture, through movements like regenerative farming and breeding with environmental impacts in mind. And so more and more growers and policymakers, researchers are all taking a closer look at some of the varieties that have come out of the green revolution of the 1960s. These varieties that were selected for traits that yield more crop, which was and remains to be crucial given the rapid increase in worldwide population. But as with any human manipulation of nature, of course, there were side effects to some of this breeding. So growers were yielding a lot more, but it came at a cost of using, in many cases, excess water, fertilizers, pesticides, not to mention all of the inbreeding that makes these varieties all susceptible to the same diseases and pests. And so one particularly interesting story that has been unfolding just over the last couple of years is that in the dwarfism trait. So dwarfism is one of the main traits selected for in agriculture since the Green Revolution. And the reason for that is because as crops were being selected to make higher and higher fruit loads, they needed to be nice and short and stout and structurally stable so that they could handle that fruit load without falling over and rotting. And as it turns out, the genetic pathway that has been targeted mostly inadvertently by breeders for dwarfism is also involved with nitrogen use efficiency. So it turns out that many dwarf varieties are quite nitrogen inefficient. So recently a research group found a gene involved with this nitrogen use efficiency that also happens to be involved with the pathway that affects growth and leads to dwarfism. And what they found is it turns out that wild rice populations have a much higher prevalence of an allele that confers higher nitrogen use efficiency than what has been selected and cultivated by breeders and growers. And so here we're looking at a map with several different varieties of rice across Asia and in the regions that they came from. And the green bar representing the prevalence of the high nitrogen use efficiency allele, and the gold bar representing the frequency of the low nitrogen use efficiency allele, and then the blue and the red regions on the map representing low and high soil nitrogen in those regions respectively. And so we're seeing that varieties with this high nitrogen use efficiency allele come from regions with low soil nitrogen, which makes sense as far as natural selection goes. And that is also being represented in this small figure down here on the right. So the frequency of the high efficiency allele goes down as there's more nitrogen in the soil. So this is what I thought was a pretty creative approach of looking back to natural selection to see how crops initially adapted to these environmental changes. And so I think that it's these kinds of discoveries that using modern genomic techniques that can help us walk that fine line of growing enough food to feed people while also taking care of the environment. And so since the first plants were domesticated over 10,000 years ago, there's been a lot of progress in breeding. And a lot of this progress, especially recently, has come from the field of molecular biology using model plant species. And while some really impressive and really major innovations have been made in molecular plant biology, a lot of this work has been done in just a few taxa that represent a relatively small portion of systematic diversity across the plant kingdom. And the problem is that there are often tens of millions of years of evolutionary time between these model plant systems. And so different mechanisms have evolved for accomplishing the same processes, from basic shoot growth to complex reproductive systems and everything in between. So here's the token slide showing the plummeting cost of genome sequencing over the last 15 plus years that we've all seen before. And it's because of this exponential drop in cost that there's been an associated exponential increase in the number of reference genomes that have been sequenced. And so these data are showing reference genomes for plants over the last 20 plus years, which are being assembled across a variety of plant families and starting to fill in those evolutionary gaps and systematic gaps. And these newly assembled reference genomes will help us fill some major evolutionary gaps in research. And Aquilegia, commonly referred to as columbines, is one of these systems. And so columbines have all the makings of a great genetic model system, especially for studying evolutionary genetics. So they're diploid, which is, we can't say about every plant. They have a relatively small genome at just about 350 megabases. All 70 plus species are interfertile, and they have relatively short generation times, not necessarily compared to Arabidopsis, but certainly compared to some other model taxa. So columbines are really well known, at least in the field of evolution, for their bread and butter trait, which is the nectar spur. And the nectar spur is a petal that has been modified into a tubular structure that at the base supplies a reward, a nectar reward for pollinators. So as columbines moved from Eurasia across to North America, they encountered new pollinators and modified their nectar spur morphology accordingly. So for example, hummingbirds encourage the development of red, yellow flowers that hang down with the spur just long enough so that a hummingbird can reach its beak in there and get the pollen, and just short enough that it will get pollen all over itself, that it will then transfer to the next flower that it visits. So when we compare Aquilegia to its most recent ancestors, these are two of its sister genera, Europhysa and Semi-Aquilegia, there's been a lot of recent speciation in columbines. About 70 species compared to less than 10 species in either of these sister genera. And it's thought that it's this nectar spur that has led to this evolutionary explosion. Columbines are reproducing more because they have more specific interactions with pollinators, and they now have this organ that facilitates diversification and pollination by different organisms, which that diversification eventually leads to speciation. So a large amount of speciation over a relatively short period of time. And excitingly, just a couple of years ago, a former postdoc in our lab found a gene that is really important for making nectar spurs. In fact, it's crucial for making nectar spurs. And that was Evangeline Ballerini, who is a mentor of mine, definitely walked me through the analysis that I'm about to present to you today for my own research. And Angie's a basketball fan, and so she named this gene Popovich. And anybody that's followed the NBA anytime over the last 25 years knows that Greg Popovich is the longtime coach of the San Antonio Spurs. So when Popovich is on, the Spurs are on. When Popovich is off, the Spurs are off. So she actually, kind of a fun aside, sent a copy of her paper to Greg Popovich, and he was kind enough to sign it and write a little note reminiscing about his experience with high school biology. And so needless to say, it made her year and made the year for the rest of the lab. So Columbine is one of those taxa that I keep mentioning, fills this evolutionary gap between the classic model taxa, Arabidopsis, and cereal crops. So this is where Aquilegia sits. And so while most of the work on Columbines has looked at their bread and butter trait, the nectar spur, I've used them to study the genetics of Alpine adaptation. I like to walk around in the mountains, and I like physiology and learning about the way that things work. And so this little plant, and it is very little, has facilitated this work for me. It's a Aquilegia jonesii, a species of columbine that is native to the northern Rockies. So it starts in the Wind River range in kind of southwestern Wyoming, and its range goes up to southern Alberta. And it lives just exclusively on exposed high elevation limestone ridges. So it's a really good taxon to be able to study adaptation, the genetics of adaptation to really extreme alpine environments. And understanding alpine environments is more and more important for several reasons. The better we understand them, the better we can manage them. And alpine environments harbor a lot of important biodiversity per capita. And this is largely because of the huge amount of environmental heterogeneity that you get in an alpine environment over a relatively small area. So you travel over a relatively small area and you get a rapid increase in elevation, and that creates a pretty significant mix of environmental niches. Additionally, alpine environments are thought to be pretty highly under threat. So as the temperature increases and plants and other organisms start to move upward in elevation to match the temperatures and environment that they've evolved to, alpine plants can't exactly go any higher. So they're highly threatened. And it also happens that, kind of luckily for me, that alpine plants happen to have some naturally selected traits that have been artificially selected in agriculture. And so there are many traits that make an alpine plant, but a couple of those that we see that are relatively ubiquitous, one of those is dwarfism. And as I mentioned, dwarfism has been selected in ag since the Green Revolution. It's also really important for alpine plants, because as alpine plants are reproducing and making flowers and fruit or cones or whatever it may be, they're exposed to really high winds and snow load and things that make it advantageous to be short and stout and be able to wait out all of that harsh weather. So for dwarfism, the genetic basis of it has actually been relatively well worked out in several different model taxa. We're studying it in Aquilegia jonesii to see if jonesii evolved the same mechanism for dwarfism, or mechanisms, there are several that have been elucidated, as what's been selected artificially in crops, or if it uses some other mechanism that's yet to be discovered. And so the other trait that we're using to study alpine adaptation in jonesii is this trait called amphistomati. So botanically esoteric term, I'll define it here. Amphistomati describes a plant that makes stomata on both the upper and lower leaf surface. And just to step back one more step, as a reminder, stomata are pores in plant tissue that allow for gas exchange, number one, and number two, the movement of water through the plant's plumbing system. So data suggests that Amphistомata is adaptive in alpine environments. A lot of alpine plants happen to be amphistomatous, so most of our evidence is correlative at this point. But the leading hypothesis there is that in an alpine environment, there's a shorter growing season, a lower concentration of CO2, and so they kind of have to make hay while the sun shines. And so having stomata on both leaf surfaces makes moving CO2 around the leaf more efficient. And as it turns out, amphistомata is also a really common and important cereal crops. That said, amphistомata is relatively poorly understood genetically up to this point. And this is the analysis that I'm a bit further along with, so it's what I'm going to focus on today. So when we go to find the genetic basis of a trait, there are some different approaches that can be taken. And regardless of what approach we take, the bottom line is that we're looking for a locus or loci that have alleles that are highly associated with a given phenotype. And that's what both of these graphs are showing in different ways. So as Paul mentioned, we can use a genome-wide association study for this. We can use quantitative trait locus analysis, and this just depends on the nature of the data, the sample size, etc. In columbines, we have the ability to do crosses, but we don't have the benefit of a deep repository of genome sequence like we do in Arabidopsis or humans, say. So we chose to do a crossing experiment with QTL analysis. And so in our crossing experiment, we crossed the species with our traits of interest, Aquilegia jonesii, to a non-alpine species that does not have these traits. So it's definitely not dwarf, and nor does it make stomata on both leaf surfaces. So crossed a Great Dane to a Chihuahua here in this case. And so I will be calling the Great Dane origami throughout the rest of this talk, and Aquilegia jonesii I'll just be calling jonesii. So I want to do just a quick review of the principles of linkage mapping here, just because it's so central to the rest of the talk. So let's just say, for the case of visual simplicity, that we're interested in the genetic basis of flower color, which of course we are in our lab, but not what we're talking about today. So we cross jonesii with blue flowers to origami with red flowers, and I've represented each of these taxa as totally inbred with these bars just for simplicity, which would be nice, but we can't actually do in columbines. And I'm representing the whole genome with just a pair of bars here, or pretending like columbines have all their genes on a single chromosome. So our F1 gets a jonesii allele and an origami allele at every locus. The F1 in this made-up case has red flowers, so it looks like the origami allele is dominant. And so we harness the recombination that happens during meiosis in the F1 to yield a mosaic of genotypes in the F2. And then we can genotype the F2 population and scan the genome for any regions where having an allele or two for one parent makes a plant more likely to have their phenotype, in this case, flower color. And in this case, we know that the origami allele is dominant. So we're looking for a region where every plant that has red flowers has at least one origami allele, and every plant that has blue flowers has two jonesii alleles. And so in this drawing, that's this boxed region here. And so we can say that that boxed region harbors a gene or genes involved with flower color, and that's what we would call a quantitative trait locus. So how do we actually go about genotyping all of these F2s? How do we know what colors go where? And then how do we make the genetic map that we'll use to do the QTL analysis? So there are several different approaches to genotyping. And as we probably all know, these approaches are rapidly evolving every year and almost every month at this point. So at this point, thanks to high throughput library prep, high throughput sequencing, and high power computing, doing low coverage or skim sequencing or whatever you want to call it is financially feasible for even relatively low budget projects like mine that are funded by little native plant societies and so on. So we ended up going with the Twist 96 prep library prep kit, which ended up being really cost and time effective. And Paul described the mechanics of how that works, so I won't repeat what he said, but I'll just say that it was really nice to be able to pool all of the samples just after this first step. And we were sequencing 5 plates worth of samples in this particular run. And so to work with 5 tubes instead of almost 500 different wells was really nice. And I took this picture here. This was a tube with one of the plates and just this tiny little pellet of magnetic beads representing the genome of about 100 individuals, hundreds of times over. So just amazing what we're able to do. So we sent off these libraries for sequencing, aiming for about 1x coverage, which based on previous crosses that we'd done in the lab, we assumed that this would give us pretty good coverage, good enough coverage to get a high density genetic map. So we got a whole bunch of sequence files back. We align those FASTA files to our reference genome, creating binary alignment or BAM files that we can work with and visualize our genomic data with a program such as IGV here. So we're looking here at sequence data from 5 different F2 individuals, 1, 4, 7, so on, each mapped to the annotated reference genome for Aquilegia at the bottom. And these blue bars are showing us just a couple of genes. So each individual has a histogram moving across this 14 kb region that we've zoomed in on chromosome 2. And this histogram is showing its coverage. So as you can see, coverage sometimes varied pretty widely across individuals and even across loci. That said, we averaged a bit more coverage than what we aimed for, with a relatively normal distribution around the mean of about 1.3, 1.4x coverage. And then when we look at individual chromosomes, coverage also varied pretty widely across loci, with a lot of variance also across individuals represented by these error bars here. But all said, and as you'll see, this turned out to be plenty of coverage to make a pretty high density genetic map. So now we've got all of our sequence and we need to actually make that genetic map. So I'm going to walk through the basics of how we did that. So after filtering our data for quality, removing indels and so on, we had to figure out which of the SNPs that we had left were actually useful for genotyping. And we used the F1 and the Jonesii parent for this. And the reason that we only used the Jonesii parent for this is because we only had deep sequence for the Jonesii parent. So we could only genotype the Jonesii parent across the whole genome, which turned out to be fine. And we didn't actually have to sequence the F1, but rather just aggregated all the sequence from the F2s, which then equals the genotype of the F1, as you can see here. So walking through this little demonstration here to figure out which SNPs are usable, the first thing is that SNPs obviously have to be heterozygous in the F1. Sites that are homozygous in the F1 got the same allele from both parents, so they're not useful. We don't know which came from which. So we go with only sites that are het in the F1. And because we only have sequence for the Jonesii parent, we need to use sites that are homozygous in Jonesii. So to demonstrate really quick, if we have a site that is heterozygous in Jonesii, we just can't say which of these two alleles actually came from Jonesii and which came from origami. Okay, so het in the F1, homozygous in Jonesii. Now we can figure out that the T must have come from Jonesii, the G came from origami, and we don't actually care what origami's other allele is. So now that we have our list of useful SNPs for genotyping, we can get started with the actual genotyping process. And so here we're looking at a table from just one individual, chromosome 1, various SNPs, and the counts for either parent at that SNP across all of our read data. So clearly 1 to 3 reads is not nearly enough to call a genotype for every SNP. So instead of genotyping at every locus, every SNP locus, we grouped these SNP loci into bins and genotyped the bins. And we can do this because recombination only happens so often, only 1 to 3 times per chromosome per individual. And so, so by grouping into these big recombination bins, we can genotype the whole thing. And those bins, we typically used about 500 kb. This did vary across the genome, and this number again came from previous crosses that had been done in our lab. Depends on the recombination rate, and as I'll show here in a bit, there's some funkiness with our physical Aquilegia reference genome. And so in areas where we had to do any manual or rearranging, we would have used smaller bins. And so where there's less recombination, i.e. closer to the centromere, we used larger bins up to a megabase. So chromosome 1, and we're looking at 18 different bins here, each at 500 kb. So then we calculated the frequency, the allele frequencies for each parent within each bin for each individual. And so we're looking at the Jonesii parent here. And so within these bins, if the allele frequency for the Jonesii allele was anywhere between 0 and 15%, we called it homozygous origami. If it was anywhere between 15 and 35% for the Jonesii allele, we called it unclear. If it was between 35 and 65% het, 65 and 85%, we said it was unclear, and any more than 85% for the Jonesii allele, we called homozygous Jonesii. So if we look at the table here from one individual across 20 different bins, we can see really clearly that it was homozygous origami through marker 1.1, and then there was a recombination event here between marker 1.10 and marker 1.11. And so we can then color each one of our individuals across every marker in the genome. And so we're looking here at sort of a pre-version of our genetic map where each column is a different individual and each row is a different locus, in this case, a marker bin. So we can pretty immediately see some problem individuals which had low sequence for one reason or another. And so we ended up removing a handful or so individuals from our analysis. Another issue that we ran into was, as I kind of alluded to just a bit ago, was that we ordered our genetic markers according to their placement on our physical Aquilegia reference genome. But as we've seen with other crosses, our physical and genetic maps don't always quite agree perfectly. So for a little bit of context, the Aquilegia reference genome was one of the last, if not the last, to be assembled with Sanger sequencing and some crosses. It is pretty high quality, but there are definitely some problem areas, especially in really repetitive regions or regions where there's low recombination. So there were some loci where in our genetic map, there seemed to be recombination events, or there were recombination events far more frequently than what we would expect from other loci. And often the genotype just wouldn't make sense given what was around it. So here's a quick example of that. We can see in this individual here, it's going from homozygous origami directly to homozygous Jonesii, which would require two recombination events within the same marker. And then it would have to happen again, just another megabase away, and then another recombination event, and so on. So not to mention, as we look across individuals, we see this similar pattern. So what is more likely than that this just being a freak act of nature, that there happened to be all this crazy recombination happening across all of these individuals in the same place? What's more likely is that our genetic map and physical map, the markers are just off. And so we ended up doing some manual rearranging based on parsimony, essentially, and what made intuitive sense. And one other thing that we did manually with our genetic map was to fill in some genotypes based on intuition. So the odds that there would have been two recombination events here, and there would be any genotype other than heterozygous or homozygous Jonesii or et cetera, is really low. So we felt good about filling in these unknown genotypes that were surrounded by blocks of the same genotype. And so we end up with a genetic map that incorporates the average recombination frequency between any two given loci. This is a more classic way of showing that genetic map. And so the average number of recombination events out of 100 between two loci defines the genetic distance between them, here expressed in centimorgans. So we can take this genetic map now and map our traits of interest to it via QTL analysis, which again, in this case, is Alpine adapted traits. So circling all the way back to our trait of interest here, amphistомati, which I'll define again because it's an esoteric word. So plant makes leaves on both the upper and lower leaf surface. So here's a microscope image showing the lower leaf surface with stomata and the upper leaf surface stomata from Aquilegia jonesii. And again, jonesii is our amphistoma species, origami is not. So here's the upper leaf surface of origami with no stomata anywhere. So we scan the genome for a region where most, or ideally all, amphistomatous F2s have at least one Jonesii allele. It turns out that there are a couple of regions. So there's one on chromosome 2 that has a gene or genes that are at least partially involved with the trait. But the region on chromosome 5 is the really exciting one. And there is clearly a really important gene, or potentially genes, under this peak involved in amphistомati. So the next thing that we go and do is look at what genes are under this QTL peak. And in this case, we just happen to get really lucky because number one, this gene is so highly associated with the trait. And number two, it just happens to be all the way out on the end of a chromosome where there's super high recombination. And so when we look under this peak, there were only about 70 genes to comb through. So not a super cumbersome task, especially when directly under the peak is a really good candidate gene. This gene called speechless in Arabidopsis that is well known to be involved with stomatal development, hence the name speechless, named after the mutant that does not make what looks like a mouth or stomata when it's knocked out. So speechless is turned on really early in stomatal development, and it's sort of a master control gene because without this asymmetric cell division that happens, the rest of the downstream processes don't have any substrate to make stomata with. So plants that have mutated versions of speechless don't live very long lives In fact, perfect knockouts don't make any stomata on their surface. So in Jonesii case, it wouldn't make sense for speechless to be totally broken. So it wouldn't make sense for there to be some major deletion in a coding region, say. And we're assuming that because number one, Jonesii survives beyond the seedling stage. And number two, we have sequence data for a lot of field collected Jonesii individuals. And there's no evidence in that sequence data that there are any protein altering polymorphisms that are unique to Jonesii. So we think it's some kind of tweak in expression, and we're going to test this. This will be sort of the last experiment that I do by trying to observe expression in vivo using a Gus reporter construct. So because the transformation technology is not yet available in Aquilegia, so we can't transform into Aquilegia, we will transform the Aquilegia speechless promoters into Arabidopsis. So we're going to take the Jonesii speechless promoter, fuse it to Gus, and make a construct to transform into Arabidopsis, and take an origami speech promoter, fuse it to Gus, and do the same thing. So if our hypothesis is correct, then, and we get the assay to work, then Gus will be observed on both the upper and lower surfaces of the leaf in Jonesii, and only on the lower side in the origami transformant. So this project is still in its nascency as far as finding the actual cellular mechanism or demonstrating the adaptive advantage of amphistомati. But I think it's an exciting jumpstart to understanding the basis of an ecologically and potentially agriculturally important trait. And so once again, I'd like to thank all of you very much for your time. Thank you for being here. Thank you in advance for any questions and suggestions you can give to me. I have a lot of both mentors and undergrads to thank that this work would not happen without. From Twist, I'd like to thank Paul Doran and Amanda Fee for facilitating this, and anyone else at Twist that also helped facilitate this webinar that I haven't yet met. And thank you to all the funding sources that made this happen as well. So at this point, I think we can open it up for questions.
Thanks, Jason, for a great presentation. It's really fascinating work. There is a question in the Q&A, and we got another one here as well. The first question is, how do you know where to start and stop each bin
Yeah, that's a good question. And there's some trial and error associated with that. And actually, I can pull this slide up too. So if you know the recombination rate for your taxon, then you can kind of decide based on that and make it so that your bins will certainly capture that recombination rate. Obviously, the smaller you make your bins, the more sequence you need, and the more computing that needs to happen. But this was one tool that we used here. So basically, anytime there are blue dots at the bottom, it's homozygous origami. Anytime there are blue dots all the way at the top, it's homozygous Jonesii. And anytime there are blue dots on all three levels, it's heterozygous. And so what we've done here with all of these little graphs is broken it up into even smaller bins. And so we use this if we ever had a question about maybe where a bin belonged or what the genotype was within a particular bin, we could kind of hone in and zoom in within a bin and say that, well, it looks like this recombination event actually, rather than happening, at the, 5 megabase position on chromosome 1, it actually happened at, a little closer to 5.1 megabases or 5.2 megabases, whatever that may be. And so that can help not only resolve genotypes, but also help us decide, if we were missing a lot of these recombination events and thus incorrectly genotyping individuals, then we would adjust our bins accordingly.
Okay, thank you. So there was a second part to that question. Would you do a binning technique to look at genetic diversity that arose through mitotic division over generations? And if not, what would you recommend?
A binning technique to look at genetic diversity via mitotic divisions over time within an organism. Well, so yeah, it depends on your question, right? So if in this case, because we have two, for all intents and purposes, inbred lines that then come together for that F1, and then recombination only happens so often, there hasn't been enough recombination to break those bins up, essentially. So that's why we can use those big bins and genotype that way. But depending on the amount of resolution that you need for your question, so if there has been more recombination, say, then you potentially would use an approach like genotype imputation rather than binning. And I just don't have enough experience with that to give much more specific of an answer than that. It's a good question, though.
Yeah. Okay, the next question is, you mentioned repetitive regions in your genome. Do you have anything to try and enrich for non-repeat or deplete the repeats?
So the simple answer to that is that we actually have a couple of new genome assemblies for Aquilegia that have resolved a lot of the issues that we have in our old reference genome. And so I know there are tools out there that can help resolve a lot of that stuff. Fortunately, I haven't had to use them because now we're able to get 20 kb reads and do things like high-C and even more than 20 kb reads to be able to resolve some of those repetitive regions of the genome and do real high quality chromosome level assemblies.
Okay, excellent. So, hey, and I was going to ask you this question as well, but Rick Nipper has brought it up. He says, "Hi, Jason, nice presentation. Joined late, so it might have already been covered, but curious as to know what kind of sequencing coverage you are aiming for your mapping population." So I think you did mention that, but my question as kind of a follow-up is, how did you determine that level of coverage?
Yeah, so again, I'm standing on the shoulders of giants in my lab that have done crosses and columbines before and determined that with a 350 megabase genome, with the recombination rate that we have, that 1x coverage is going to give us a perfectly dense genetic map to be able to get down to a region. For a more general answer, it's of course is going to depend. It depends on the size of the genome and really your question. So obviously the more dense your genetic map, the tighter you can hone in on a gene. But if your budget is such that you can only sequence 300 individuals, or any logistics are that you can sequence 300 individuals like it was in our case, then that's a low enough sample size that you might not even get enough recombination to narrow in all that closely on a gene. And
Right.
So at that point, it could make sense to save some of your sequencing ability for fine mapping further down the road. And that's where having these whole genome libraries is really nice, because we have the library for the whole genome. We can always go back and do more sequencing if we need to.
Okay, great. Question from Beatrice Landoni. Have you used any particular bioinformatic tool to clean and handle your low coverage data, which I think is great, because I think you're actually working on a full suite of tools.
Yeah. So bioinformatic tools to filter the low coverage data.
Mhm.
Yeah. There are a million tools out there. And the one that I've have the most experience with is GATK. Um, and things like SAM tools, VCF tools, and honestly, I've kind of used a smattering of all of them to accomplish what I need. In this case, for this mapping experiment, there wasn't all that much filtering that needed to be done. And we could chop out a whole bunch of SNPs without having a huge effect on the downstream analysis, because we just had enough to be able to call genotypes within these 500 KB bins. But that obviously becomes more relevant if you're trying to do something like impute genotypes. So I meant to plug it in here and I didn't, but I'm involved with a group right now that has developed a tool that's geared toward non-model systems. And as this technology is evolving so rapidly and becoming financially feasible for those of us doing ecology and evolution, then it's important to be able to figure out what to do among this sea of different techniques. So it's called marine omics. If you Google marine omics, then it'll come up. We have a GitHub repository and a GitHub web page where we're starting to aggregate some tutorials and tools for being able to do things like everything from filtering data, experimental design, all the way through analysis and interpretation and so on.
Excellent. Yeah, so I see this marineomics.net, marine molecular biology and genetics, proteomic tools, transcriptomics, genomics. Yeah, so this is...
Sorry, Paul, it's actually marineomics.github.io. There's another marineomics out there.
Okay, yeah, marineomics.github.io. Got it.
Yeah,
okay,
that's the one.
All right, so we got time for one more question. The question is there a way to detect historical recombination using NGS? Let's see, the question got bumped. Using NGS data. Is there a way to detect historical recombination using NGS data?
Historical recombination, as in recombination that happened before this cross in either of these taxa, I suppose is the question.
Yeah, I think yeah.
So I don't have personal experience with that. And so unfortunately, I've just got to say, I don't know.
That's perfectly fine.
Yeah.
So the last question is actually perfect. It says, thank you for the great presentation. Can you share the recorded meeting? We will be posting this on our website. And so I think Amanda might be able to mention when and where that will be available. But just one kind of last comment, a quick reminder, we're going to ask for a brief survey. So if you have a minute to spare, we'd love to hear your feedback. Jason, thanks so much for the fantastic presentation. Obviously got a lot of great questions, and thanks everyone for coming. Have a fantastic day. And like I said, we will definitely post this event on our website, and you'll be able to watch it anytime. Have a fantastic rest of your day, night, or afternoon.